Hi, everyone. Thank you for being here today. Dr. Lance Waller is our first IDAS seminar speaker for the spring semester and for 2025. So you're all probably familiar with him, but I'll just introduce him a little bit more formally. Dr. Waller is co-host of the IDAS seminars. He is an IDASTP steering committee member and an IDASTP training faculty member. He is professor in the Department of Biostatistics and Bioinformatics at Rollins School of Public Health, with a joint appointment in the Department of Environmental Health. His research involves the development and application of statistical methods for spatially referenced data, including applications in environmental justice, epidemiology, and disease surveillance, to name a few. Today, he'll be giving a talk entitled Questions, Data, and Maybe Answers, Research Challenges When Spanning Scale. Thank you, Dr. Waller, for being here today.
Great, thanks, Nicole. So I traditionally give this first talk. Some of you have seen it before, but I hope there's some, I will be using some different examples. And I thought I might give a little bit of an introduction of myself on the time scale because everybody thinks I just turned 60 years old yesterday. So that's given me pause to think about things. When you turn 50, you think, oh, I'm halfway there. But when you're 60, you're like, I don't think there's a lot of 120-year-olds out there. So um, when I was department chair for biostatistics for 9 years, I made a list that I would share with graduate students. And I hope this is helpful to think of. Several people have asked for copies of it from time to time. And so these are things I think it's good to keep in mind. And so first one is read, read, read. I think you should read stuff in your area, but also other areas. A lot of times I would go to seminar or go to conferences and I'd go to all the talks about what I was working on. But I had gotten into the habit at one point of trying to go to a session I didn't know anything about just to see how this works out. Yeah, thanks for the happy birthdays. Uh, And then the second one is really the same, write, write, write. One of the big parts of a research career is being able to write, whether that's an email that you write in a way to get a response. And believe it or not, there's ways to write emails that don't get a very fast response. So it's too long, or it includes too many people, or something like that. Or it's writing research papers. And as you read, you'll find people that describe things that you like the way they write. There's different writing styles. You'll learn from them. Their teaching style will line up with your learning style really well, and help go iterating between these two help you find out. Like I'll know certain researchers, if they wrote a paper about a certain topic, whether it's a method, I would like to read what they wrote because I like the way they think and describe things. I find it really helpful. They may not be the first person to write it or the leading person to write it, but if they've given it some thought and describe it, I sort of find like I understand their language. Um, Also, you should learn from good and bad writers and presenters. So if you're at a seminar or a conference presentation, you're thinking, this is just terrible. I don't know what they're talking about. Start paying attention to why it's bad and then don't do that. I've seen some pretty bad presentations, but if I start paying attention to like what is annoying me about this, or why am I lost? Are they not defining things? Is their notation bad? So you can learn from counterexample by bad examples too. I mean, don't seek them out, but don't just dismiss them and stop paying attention. And then when you find the people whose teaching style lines up, you kind of identify those as your heroes in the field. Those are the ones whose work you read other things they've done. Because again, it's sort of like you're speaking their language and it helps out. And then when you're working on your research, it's really helpful that you should like at least part of what you do. If you're just doing it because it's an assignment and was the next thing to do, then you're not thinking about it when you walk your dog, or you don't get excited about explaining to somebody new what it is you do. So it's really important, I think, to keep in mind that even when your experiment's not working, or the deadline's coming up too fast, or there's still something that you're kind of like, hey, this is kind of cool that I work on this. And I think that's important. It's also good to keep a hobby, do something to kind of clear your mind. I can't tell you how many take-home exams in graduate school I solved the day afterwards because I finally did something else. I wasn't just keep rethinking the same approach to a problem and running against the same brick wall. And sometimes it's the creativity that comes out of doing something else. And I'll give an example of my hobbies in a minute. And then use your resources. So your advisor and your committee, they're a resource for you. They're not the final gatekeepers. They play that role, but they're also there to answer questions, put you in touch with people. Your director of graduate studies, the administrators who know the rules, your senior classmates. These are all things that are really important to do. Know the requirements and meet them because not everyone on your committee is going to remember that there's a form for this exam or something like that. Just remember to check in on those from your resources. And the small world, after all, part is it's it seems like there's a million scientists in the world, but once you narrow down to your field, there's only a handful of them. And the advice I got indirectly in graduate school, George Casella told one of his students who was a classmate of mine when he was going out to interview, he said, the field of statistics is really small. Don't piss anybody off. And sometimes we try to show what we know, and maybe your advisor didn't like somebody at a place you're interviewing, but they all know each other. And just because you go somewhere and interview and don't get a job this go around, 5 years from now, 10 years from now, 20 years from now, you might be looking at the same place with the same people. So don't burn your bridges. Then the imposter syndrome is a real thing. I'm sure you're all aware of that. But you need to, no one goes from knowing nothing, you're learning stuff in your field everybody knows in the beginning to stuff only you know at the end. In 5, 6 years, that's a really short amount of time. And there's just no way that goes perfectly smoothly. And everyone will hit this stage at a different frame of, I don't know what I'm doing. Everyone else seems to know the next thing to do. They all have their act together. That's just part of doing research is that we don't know the answer, and some things we try aren't going to work. So you're not going to be alone in this. And then this last one kind of sounds like a haiku or something, but I came up with this about a year ago. You should try often. You're going to fail sometimes, but you should learn each time, whether it's success or failure, just like good or bad. Um, then I wanted to say with the hobbies, like I like to read, and I've read Frankenstein several times. I really like the book. It's not a big scary monster book, but Mary Shelley wrote in there that if you're imperfection, a human being in perfection, ought to always preserve a calm and peaceful mind, never allow passion or transitory desire to disturb your tranquility. I do not think the pursuit of knowledge is an exception to this rule. If the study to which you apply yourself has a tendency to weaken your affections, to destroy your taste for those simple pleasures which um, no alloy can possibly mix, that's to say not befitting. So if you're only doing your work and you're only doing your research, you're missing out. So what do? I like to read, so that's where this came from. But also, so I've been thinking like I'm 60, what did I do each decade? In my first decade, I learned how to read, and I thought that was good. But I also, clearly had a performance bent. The second decade, oh, that looks like the same hat, but it's not. This is me in high school when I got a guitar, and this is 1980 here, the first electric guitar, and then I still have the Fender Stratocaster that's over here, and two different bands I was in. The third decade, I went to graduate school, and I couldn't find a picture of me playing guitar. I played, but not as often. This is me in a probability class, and the guy next to me has already received his grade, but we haven't gotten to Waller yet, or I would be doing the same thing. That was not my top performance. Fourth decade, so my kids were small. Here I'm demonstrating. Oh, look, there's that guitar again. At like parent day. Fifth decade, here I'm playing with some people. I'm wearing my famous turtle nerd shirt. Oh, that's the same guitar again. And then in my 50s, oh, I got an additional guitar. So I still have the Fender Stratocaster, but I also have this pretty blingy Gretsch. And I'm wearing my math shirt here with pies and infinities on it. And then I stepped up my game, and there I am at tuxedo playing at my wedding reception a couple months ago. So the evolution of, clearly I have a performance bent here, but I'm still the same person, just 50, 40 years later. So there's the collection. There's the Stratocaster. Yay. There's the Gretsch. There's two acoustic guitars and then a cool Giannini Craviola 12 string. So that was kind of an aside. It was self-serving, but I was thinking the other day, when I turned 50, I go, what did I do each decade? First decade, I learned to read. Second decade, I got into music. Third decade, I got a job and got married. My kids were born. I was a parent. And then we'll see what I do in my 60s. So I can hear the wild applause. Thanks for your attention. Now let's talk about scales. Or first of all, are there any questions? Not that I expected.
I have a question, Lance.
Yeah, okay.
Your 12 string, can you play different scales on that than on your 6 string guitars?
Not that one. It's hard because that one actually has a really narrow neck. So the 12 strings are really close together. So
Mm.
basically you can strum. It's very hard to finger pick. I've played guitar with the op before. Those of you who've been on the retreat know that. But it looked cool because it had the weird body shape. And I bought it. It only had 6 strings. I bought it for $50. Thought it would look good on a shelf if it didn't sound good. So it's been on a shelf ever since. So it just goes to show you try often, fail sometimes, learn always. But I did get it fixed up so I could play it. All right, questions, data, answers. This is the talk where we think about where the cross scale aspect comes from. So let's see what we're thinking. So this course is a seminar course, but it's a course about ideas. So each seminar speaker will be thinking about their own research and ideas, but we also want to think about them collectively. That's one of the reasons we do the training grant and the training program. So we have a course on hypotheses. It's a course on data. We're going to see people using different types of data and answering really interesting questions. And it's a course on how they do their research. And it's a course on questions, the questions that drive their research. These are framed as hypotheses, but really they have something behind it. How do butterflies navigate? How is Lyme disease spread? How could we break the cycle of transmission through these interspecies things? And it's a course for discussion. So you'll see people asking a lot of questions in the class, and we encourage you to do so. So if we think about how we learn things, whether it's research or not, we have questions we want to answer, and we're going to collect some information to answer those questions. And then there's data that we do collect, and we have methods to analyze those. And then we have questions that we can answer with the data we get. And those of you who've been here before know this picture. So the virulent vortex got its name from the swirling vortex analysis. When I first moved to Emory, in 1998, I went to visit the EPA Region 4 office downtown, and one of the toxicologists there, John Richardson, we were talking about solving problems with geographic information systems, and he said, it's just like this, and he drew this diagram on a piece of paper, and I said, oh, can I use that in class? And he said, sure, and I've been using it ever since. So it's really the scientific process bent in a circle. This is a question you want to answer in the upper left. Before you do anything, you could say, what data would help me answer that question? And you dream big. I would like to follow everybody all the time, all the animals. I'd like to know where the ticks are. I'd like to know where the mice are. I'd like to know where the deer are. I'd like to know where the infected ticks are. I'd like to know how people hike through this zone and get ticks themselves. But then there's the data I can really get. I really can't follow everybody all the time. So what data can I observe and what scale do I see that in? And the data I get and the methods I use give me some answers, but they don't answer everything. So there's the questions you can answer with the data you do get at the scale you observe it, and you're trying to decide, this last step is really important. How close is that to the question you wanted to answer? What part of the question do you want to answer is that? So you'll go around and around. So the next thing you could say is, well, how could I get closer? What data would I need to get closer? Let me see which data I could find that are like that, and let me refine my answer. So the scientific career, we just go around and around. And I've been using this for almost 30 years to teach. And just last year, someone said, I don't like it. It sounds like you never get anywhere. So maybe you think of it as a spiral staircase, and you're always going up. But there's no single study is going to finally answer the question. I think there's always the next time around the cycle. So how does scale fit into this? So something like a scale is, there's a scale at which a phenomenon happens. There's the scale at which a tick bite transmits ehrlichiosis, or a vector transmits a pathogen, or I sneeze and particles in the air get inhaled and you get infected and we both have the flu. So it's the scale, and then there's the scale at which phenomenon's observed. I may not follow every tick every day. I may only see the people that come in with ehrlichiosis and I found out where they live, and then I ask them where they went hiking. But I may not talk to the people who never got ehrlichiosis and where they went hiking. So there's a mismatch here. And there's also the scale at which a phenomenon is controlled or adjusted. So we've been seeing in the news a lot about vaccine coverage rates, and we might think about where vaccine coverage rates are higher and lower. And then where is a phenomenon controlled, adjusted, or less controlled or adjusted? If you lift requirements, what does that change things? And these are not the same. So I have a rhetorical question. Are these the same? But they're not. And sometimes there's the way you can observe it or the way you can control it. If budgets are allocated to county health departments, then monitoring a county level number of cases is helpful. But if the budget's only allocated at the state level, then maybe that's more detail than you need. So with infectious disease, as we're thinking about infectious disease across scales, we have the scale of the host and how the host moves. We have the scale of the pathogen. What does the world look like to the rabies virus? We have the scale of the vector and its movement. So if it's a vector-borne, how does a mosquito move? How much does it move during the day, in the morning, at night, within a room? Like David's been looking at tracking individual mosquito movements inside the same room. How high do they go? Do they react to different color paints on the wall? Scale of the environment and its impact on the other scales. If temperature goes up everywhere, how does that change? Are we more at risk for vector-borne diseases as average temperatures go up? What's the scale of contact required for transmission? Is it a single bite, or is it by an infected mosquito, or is it repeated exposure? And then we also have the scale of observation and the scale of control. We can keep going with these lists, but you can start to see the mental exercise here. So I want to do a couple of examples. So we might think about flu. You might think about what's the global scale of flu, what strains are circulating in a given year. Same at the national scale, the state scale. If you follow the morbidity and mortality weekly report, when the flu season starts in the fall, there's a lot of testing going on to figure out what strain is circulating and whether it matches the vaccine that year or not. And then within a given state, there might be a county scale. Within a county, there might be a city scale, or across counties for the city of Atlanta for a large metropolitan area. Might be interested in the neighborhood scale or at a school. And within a school, there's classrooms. And within a classroom, there's kids' hands and noses, and there's sneezing, and there's shaking hands and sharing pencils, and so on and so on. We can go down to the molecular level of how the actual virus is working. There's also time scales, the annual scale, the seasonal scale, the monthly scale, the weekly scale, and the daily scale. Every one of these has interesting questions in it, but to do a study at any one of these might look different than a study at a different scale. We saw a lot of examples with COVID-19 about monitoring it when it's first starting. Global scale, national scale, state scale, county scale, city scale, neighborhood scale, home scale. If you remember when new strains would start and the first case in the United States was in the middle of Kansas or something, you know it probably has to be everywhere already. If it's already coming in through New York or San Francisco and spreading across the country, it's just the scale at which you see things, it may not line up to the scale at which it happens. And then there's the individual scale, and the immunological scale, the virus scale, the protein scale, the genome scale, all the way down. And then what scale of response should we have? We don't want to have a national genome scale response. That would be, when we talk about personalized medicine, though, for cancers and things, sometimes we do talk about gene level responses and trying to customize treatment. We might think about how effective is that for individuals? How effective is it for groups of individuals? And what's the overall cost? Measles, we can think of the scale of outbreaks prior to vaccination in the early 60s before 1964 or so. And then the scale during near full vaccination, maybe 7 or 8 years ago, 10 years ago. And then we can see as some of the vaccination rates have dipped down a little bit, what is the scale in terms of coverage? There was a outbreak at Disneyland one year. There's the scale at Disneyland at which individuals may be infecting one another. What about the scale of the outbreak for people who weren't there that day? I want to think of another example here, and I'm going to stop sharing for a minute. First of all, let me get any thoughts you might have on scale. And then Natalie Olson, who's in the class, and I'm one of her co-advisors, is going to show an example of an MPOX study. She's working on a mathematical model we're trying to develop to look at land use change and animal to human transmission of mpox. So let's start thinking about those sorts of things. But maybe David, do you want to say anything about the mosquitoes?
Uh, can y'all hear me?
Yeah.
Okay, sure. Um, yeah, so I guess in my case, when I got thinking about how do we consider the behavior of the mosquito to improve target control or, yeah, vector control, I pretty much was exposed to how, you know, targeted indoor residual spraying came about, and that got me thinking, which for those of you who don't know, targeted indoor residual spraying is just spraying the walls inside of homes at a particular height. In the case of mosquitoes, it's 1.5 meters. And that got me really thinking about kind of what you mentioned about the movement of the mosquito and kind of some of the observations that uh, people were seeing in the field. And when I started kind of looking deeper into it, it was pretty evident that not a lot of research had gone into trying to understand what patterns mosquitoes display in terms of not just flight, but also resting. And that's what really got me thinking. And thankfully, my lab has access to this very fancy machine that we're having, thankfully, the opportunity to beta test it. And that allows me to really get at some high resolution. So looking at mosquito flight at much more detail than before. And so that also kind of brings in the scale aspect because now you're just looking at coordinates in space and time, and you're looking at multiple individuals. And how do you take that into consideration when you have 100 individuals? Like, what does that tell you about the population, quote unquote, flight? And so you can really start thinking about many different things when it comes to scales and how it impacts your vector or your disease or your host, whatever it may be.
Great. Thanks for being on the spot too.
Of
Like you
course.
said, one of the aspects of the scale of treatment there is trying to concentrate the spray of the pesticide to the, 1.5 meters down the lower part of the room so you don't soak the whole wall floor to ceiling. That reduces the, incidental exposure of the humans to the pesticide, but it also is much faster to do. And if it's equally effective, you can cover more houses and probably have more of an impact on a neighborhood or community scale. So you can think about what you're having a policy that's based on an individual scale behavior of mosquitoes and saying they seem to rest underneath the tables or on the lower part of the room. David's measuring whether that's really the case, and then also how effective would the pesticides be at if you only sprayed part of the room. And if you could get 80% of the mosquitoes but only spray half the room, then you're ahead. You can do almost twice as many houses. So thanks, David. Natalie, do you want to share? Do you have that? We were talking about that map you have. Yeah, great. So why don't you tell a little bit about the project, and then we can get some comments from others. And others, please start adding stuff to the chat. That way, A, we know you're here, and B, we can tell which jokes are landing and which ones aren't. All right, thanks. Go ahead, Natalie. You don't have to have jokes, Natalie, if you don't want to.
Oh, I always have jokes.
Okay.
Probably not right now, actually. But so yeah, when I was kind of getting started on figuring out my dissertation aims, I was primarily interested in zoonotic spillover and thinking about how disease is transmitted from animals, like an animal reservoir in wildlife or livestock into humans. And one of the things that I kept coming up across was it was kind of taken as a given, and was repeated really often in the literature that land use change and deforestation were really important triggers for zoonotic spillover. And so every time I would read that, I would kind of think, okay, that's really interesting, but like what kinds of land use change or what kinds of deforestation? Because that is a process that can happen in a lot of different ways, depending on what it is that people are doing with that landscape. And so we decided to take the Congo Basin as a sort of case study, because it is an area with a lot of different extractive industries happening, with a lot of different countries. There's 6 countries with kind of all different policies, and with a lot of forest cover that's kind of being deforested or affected by a lot of land use change. So we kind of wanted to ask how land conversion trajectories might be different between different extractive industries like logging or mining, according to our model. So we ended up creating this mathematical model that simulates these different land use patterns to compare the probability of, in this case, MPOX spillover between the different sort of paradigms of land use change.
Yeah, so you could think about the scale of mining, what the scale of landscape influence. How does local mining change the landscape that could have impact on animal-human interactions? How does forestry or logging concessions change that? Do you want to, can you say like what's different about that? Like what kind of landscape changes do you see for forestry and what do you see for mining?
Are you asking me or the broader group?
Well, both, I guess. So first of all, the broader group, why don't some people put some ideas? What's different about mining and forestry in terms of scale of impact? Anybody can jump in. Oh, I have another 12th thing for grad school success. It's offer ideas. Well, Natalie, do you want to fill them in? Oh, there you go. Mining probably has a longer term impact in terms of people being there more long term than compared to logging. Yeah, that's a good point. Natalie, you want to comment on that and that you've looked at it before?
Yeah, so that is a really important part, is that you have a lot more people kind of migrating into mining sites, not just for mining jobs, but then also because there becomes this developing economy around an established mining site. But really the primary difference we kind of think of is with logging, you have logging activities that are moving through the landscape at a faster pace. And they're only like selectively logging usually, so it's a little bit less severe of an effect on the landscape. Um, and then it's usually along pre-established like road networks or recently developed logging road networks. So it's going to be more of like a linear and branching spread. And again, it's faster but lower severity. When we think about mining, usually they will kind of discover a deposit of some sort of valuable mineral and establish a mine there. And what often happens is kind of secondary, what they call artisanal mining sites will kind of pop up around there because the infrastructure has been kind of set up to be able to then sell kind of those minerals in that same area. So you have kind of a slower sort of spread of mining activities from that sort of central site. But the degradation of the environment due to mining is a lot more severe. You have to clear cut to kind of establish your mine. And then there is also a lot of degradation of waterways. So again, it's kind of more severe, but it's much slower spread. Usually stays pretty co-located around that central established facility. But yeah, so those are kind of the main differences that we identified.
Yeah, thanks. And we also might think that mining, by being in a place longer, may end up developing these sort of temporary or longer term temporary housing or collections of workers. Then you have to have the support industry, the people cooking food, and maybe that turns into restaurants. Maybe there's sex work, and then we have to think about mpox transmission that's different from animal to human. And so over the time scale, these interactions and transmissions, even though it's the same disease and pathogen being transmitted, is changing over time. And how long you're staying in a place and how crowded it is, and all that's going to play into this. So you can see it's a pretty challenging thing to model and to come up with tracks. So thanks, Natalie, and thanks, David, as well. Let me go back to some of my presentation. So going back on earlier work, so this is even older than me. This is John Snow's cholera outbreak in London in 1854. This is a different display than the map you usually see, but it's a graph of cholera cases and diarrheal cases, so they line up pretty well over time. And then in the background are all these meteorological things like temperature, and then there's a smoothed version and how much rain. And they were kind of doing the same data sciencey things we do now, except we have thousands of variables. What's the magic signal that happens right before this goes up? Now, if you don't know how the causal mechanism works, like they weren't, the Vibrio cholera had been seen in a microscope, but not connected. And they were, going off of the air was bad, or as a combination of temperature, and you could smell the the filth in the air, and you worry about that. Oh, I need to share my screen again. Can you see it? Okay. All right, thanks. Anybody else not seeing it? It has a little green line around it, so I think we're good. Because you don't want to miss this. There's John Snow looking at the map. So this is the picture of John Snow. We have a map of cholera deaths in London, and the dotted line goes to the Broad Street pump. And he was thinking, oh, the cases are concentrated here. It must be the pump that's causing it. The other X's are other water pumps. But Snow did more than just make this map. He also interviewed the families at all these, the residents. And there were some deaths that were closer to other pumps, but they were getting their water from the Broad Street pump because it tasted better, or that was the one they were familiar with. And yeah, I've seen snow out of the window too. And so they kind of gave us neighborhood scale. Oh, which ones are closer to this pump? That drives it, and it's kind of the beginning of modern public health. And Snow's stuff is really amazing to read if you haven't read any of it. But does identifying a scale always lead to the correct conclusion? Well, not really. At the time, the London Board of Health met and saw that's the problem of having the chat open, is there's always a pool running on can we throw him off his tracks. Parks was a member of the London Board of Health in the more traditional, and he said this certainly looks more like the effect of atmospheric cause than any other. And the paragraph before this one, he goes, so Dr. Snow has shown us that there are more cases around the accused pump, concentration, almost a circle, and you're kind of like, yeah, you're going to take the step and say it must be the pump. And he said, no, it must be the air, because if it was just the water, why isn't it around every pump? So he was thinking the water is, his scale of water was water's the same wherever you pump it out of, as opposed to there might be something particular about the Broad Street pump. So the two of them, and they're both scientific thinkers, are looking at the same data, the same map, and they draw different conclusions because they're kind of bringing their preconceived ideas of scale and assumptions to this. So Snow's success, he did win the day, was not only due to the map, and in fact, even seeing maps, this is an earlier, I like disease maps. So this is a map of yellow fever in New York City that a gentleman named Valentine Seaman did in 1798. He said, oh, there's mosquitoes in this area, and it seems like the cases are by the mosquitoes, so mosquitoes also must come from miasma. So the circumstances favoring the rise of the putrid miasma, the bad air, equally favor the generation of these insects. So he did not say this in 1965, he said it in 1798. But they're seeing a pattern that, from our perspective, shows the lineup of the scales, but it doesn't. So even when the scales align, we have to remember and carry some humility that we carry expectations and assumptions that can cloud our understanding and interpretation. And then, are we just seeing what we wanted to see? And as scientists, we need to ask ourselves that all the time. What are some other possible explanations of the patterns we're seeing and the conclusions we're drawing? Scales can change what we see. And one of the things that we see a lot of times if you take an epidemiology classes, they talk about the ecologic fallacy, or if you take a geographic information system class, they talk about the modifiable area unit problem. And if you have, this is two maps of the same data, it's just that I've aggregated it for the map on the left. And so where the dots are, where the cases are, have some patterns at small scales that are completely lost at bigger scales. And so, and in this concentration in the middle has a whole lot of dots, but it doesn't look as scary over here because my boundaries go through the middle. And this is how gerrymandering works. If the dots are the people voting one political party and the other areas has the voters from the other party, cracking is where you try to draw the voting boundaries like they did in Athens, Georgia. They kind of split a liberal city into the little parts of including a lot more rural Republican areas. So a more conservative total vote by splitting up the liberal vote into smaller pieces. So this is why why gerrymandering works, but it also is kind of why there's no single solution to this, because no matter how you cut, you can start to change your answer. I also tend to think of spatial filters. And so in disease ecology, if all of these are maps, if you have a map of the hosts, where are the mice? Where are the ticks for Lyme disease? Where are the pathogens? So the infected ticks. These maps all interact with landscape and climate in a dynamic way. The animals are moving around, they're infecting one another. And all together, they generate this marquee map in the middle of the true cases in the host. That's the map I really want to see. But I don't get to see that because if these are people, they have to go to doctor, the symptoms have to be diagnosed. Maybe the doctors in the north part of my map are really good at diagnosing and 100% of the true cases show up. Maybe in the southern part, people are more spread out. The doctors aren't as quick to diagnose. So individuals come in and only half of them show up. Now the map here is going to be a different spatial pattern because of the filter it goes through. Similarly, the doctors in the north might be really good at reporting cases to get to the CDC or the state health department. And so the database with the reported cases has all those. And if the southern one, the southern part of my map is slower to report, then that could thin that even more. Or if vice versa, if the southern part is better at reporting, even though the diagnosis was lower, this may be low. And then the top northern part may have, even though they diagnosed all the cases, they didn't report them in a timely manner. And in public health, we usually start on the right here and try to work our way back to this map. But we need to understand what are the local factors that filter the results. So the scale at which things are observed, how they're observed, and where. So with scales in disease, we have scales of hosts, scales of parasites or pathogens. We have coevolution, like the pathogen may go through multiple generations in the lifetime of the host. And you can end up with some changes there. The molecular clock may be moving very differently. And can we change scale with control measures? If we have effective control measures like, say, vaccination coverage, we can start thinking about how that map changes the map of transmission. So a good way to start this class is, so this is a pretty old paper, but also from a very classical ecologist, ecological theorist. Simon Levin gave the MacArthur Award lecture, and this is sort of one of those career type talks you give. Looking back, here's my view on the big picture. And he talked about scale and pattern as core concepts in ecology. And one of the challenges of ecology is we view things happening together and we're trying to infer how all the processes work from those patterns. So 1989, so I realize this is a long time ago. We can age this by which guitar I was playing. He published this paper in 1992 in Ecology. It's got, I'm sure by now, more than 4,000, well above 4,000 citations. And it's a really good place to start. So we try to, I assign this at the beginning of this class every year, not to grade necessarily what you turn in, but to have you read it and start thinking about it. And we think it's helpful to do this at the beginning and the end of this class. It's a good paper to read multiple times. Some of it's dated. When he talks about computing and doing things 10 times, it's going to sound very quaint compared to what you can do now. But he called it the problem of pattern and scale in ecology. And here's some quotations I'm going to give. We're just going to break down the abstract. So I encourage you to read the whole thing to see how he plays this out. He talks about the problem of pattern and scale is the central problem in ecology. And I'm highlighting certain areas or bolding certain. So the central problem in ecology is a pretty bold statement. But he's saying seeing patterns and the scale at which patterns occur is what generates hypotheses, and it drives the design of our research and data collection. And so it unifies as population biology, just talks about how things grow, how populations grow, how resources limit them. Ecosystem science, how, say, the rate of growth of plants growing and fertilizers, natural fertilizers, these different things. And it marries basic and applied ecology. So this central thing of observing how things happen and at the scale that they happen drives a lot of our thinking in ecology. So Jaap and I think we agree with this is the central problem in ecology because it's the central problem for our training set. Now this is again all from the abstract. Applied challenges such as the prediction of ecological causes and consequences of global climate change, and this is from 1989, require the interfacing of phenomena that occur on very different scales of space, time, and ecological organization. We'd like to include in ecological organization the impact of control measures. So the Paris Climate Accord, again, is in the news after yesterday. We can think, what can countries do and how does it change space, time processes happening and at what scale? How fast do things change? How much do they expect them to change? And it's the interfacing of these things that happen, and you may change something at one scale and it has implications for another scale. And I think this third one's really important. Just like I was saying for gerrymandering, we see why it works, but that doesn't give us a solution how to prevent it from happening. And some cases it's very egregious. In some cases it may be less so, but it still benefits groups of others. This is important. There's no single natural scale. I'm not trying to find the magic scale at which a phenomenon should be studied. Systems generally show characteristic variability on a range of spatial, temporal, and organizational scales. So if I go back to my whirling vortex, if your question is at one scale and I'm collecting data at a different one, I may get the right answer, but it's not the right answer for your question, or it may not even have that much direct influence on the answer at your question. We hope it's all it all interacts and there are processes working. It's just that the scale of observation can impact the conclusions and what we see and what we think. And that aggregation effect has, not only is it sort of insidious for voting districts, you can also think about saying whether a program's effective or not. If it's effective at a 5-year scale and you only show the 1-year result, you can say it's not working, we're going to pull the funding from it. Sometimes, again, that's intentional. Sometimes it might not be intentional on a political scope, but we need to think about the fact that measuring things at different scales may give different answers and draw different conclusions of whether something's working. It may be working at that scale. That context is really important for disease ecology, for environmental regulation and policy, and for health response. And from my filter before, this is, this Levin paper influences my thinking a lot. I don't know that I realized it as much at the time, but now these things stick out as you come up with ideas over your career. The observer imposes a perceptual bias, a filter through which the system is viewed. That might be intentional, like the design of your data, the scale of your data, but it also might just be what you're used to looking at. So we had the London Board of Health, and then we had the yellow fever in New York. There's a filter by which they were viewing this, and it has fundamental evolutionary significance since every organism is an observer of its environment. So measles is an observer of how many unvaccinated children there might be, or susceptible children, is probably a better way to put it. And life history adaptations such as dispersal, dormancy, alter the perceptual scales of the species and the observed variability. So, okay, thanks. It also has fundamental significance for our own study of ecologic systems, since the patterns that are unique to any range of scales will have unique causes and biological consequences. So one of the running jokes, Jaap and I get teased that we just keep bringing up that it's a scale issue. You should think about the scale issue, but that's almost the point of our training program. All the skills you learn in the other courses all operate at specific scales, and it's important to be aware of those. there's the spatial filters I had. So I was feeling totally justified that this came up again, 20 years before I actually wrote it myself. So the key to prediction understanding lies in elucidation of mechanisms underlying these. We'd like to use patterns to understand processes, but that's not a unique connection. So the patterns, we try to implement that sometimes through mathematical models, generate patterns, see if those look like the patterns in the data, and then go back and forth between pattern and process. But typically the mechanisms operate at different scales at those than which the patterns are observed. That's one of the hard parts, and that's having awareness of this, I think, drives us digging deeper into our research problems. Another way to say this is the scale at which things are observed provides a context for interpreting and the generalizability of your conclusions. So that ever-present idea, there's a scale associated with what you did, and that scale has impacts on the conclusions you draw. Once you know that, then I think you can be more specific about your conclusions. In another case, the pattern is imposed by larger scale constraints. And we could think of examples for this, but given the snow and all that, I'd like to give some time for discussion. The other paper that we can put up is looking back on Levins. And I think Chavez's paper is a really interesting one. And it basically talks about how Moore's law computers got faster and faster. We have all kinds of measurement techniques, which also operate at scales, PCR, genomic sequencing, single cell. We have environmental sensing. We have monitors and lots of them. This is from 2013. You can think about all the scanners and wearables and stuff we have now. And because these all generated lots and lots of data, we have information, data science, and up to AI. We have ability to disseminate results on a global scale. And as we saw with COVID in 2020, some of that dissemination is immediate through preprints, and some of it's accurate and some of it's not. So there's a lot to discuss in our seminar series. So listen actively when we've got people talking, start thinking about what's the scale of their problem, what's the scale of their data, what's the scale of their observation. Sometimes we tend to backtrack and introduce our results with the scale at which we could operate, and as if that were the only one. Think actively, write actively, suggest actively, just participate in the discussion. Here's the homework, yay. So I'd say read steep in some teaching literature. They talk about interrogating the literature you read. So, read the Levin and Chavez paper, focus more on the Levin one and sort of think, what can I take away with it? Or what doesn't make sense anymore? Or how does this fit? And then, I'd say give yourself 2 to 5 pages, summarize the key points about scale as you see them, and talk about an ecologic system you've read about or you're interested in or you're working in, and think about what are some scale things you should keep in mind. And then, kind of read it again or read what you just wrote. What did we learn in the 10 additional years since the Chavez paper was the 10-year check-in? And then reread it, revise it at the end of the semester, look at it again. We've had people do this and they've redone it every year. I'm not saying that it was a brilliant homework assignment, but they said they found it helpful because they're at a different stage of their work each year, and it's good to check in on this. So if you could bring that, give it some thought, bring it next week. You're not going to get really detailed feedback and stuff. Yeah, it's homework that scales across years. Thanks. Yeah, we're tracking longitudinal. And just revisit the ideas, because this is kind of the things that we want you to take with you. And then remember this picture. You've got questions you want to answer, data you would need to do that, data that you can get, and then the interpretations analysis you can get at those scales. So I hope you find this helpful, at least for framing and thinking things forward. Yap and I, now we've just totally absorbed that this is how things should be, and we think it's a good idea, but now I'm interested in, the next 5-10 minutes, other thoughts you might have or examples you might give, or, concerns, questions you might have. Any thoughts? You can put them in the chat or you can unmute.
Gonna maybe say one thing that may also help discussion is that when we first, wrote this training grant, we had really different ideas of what scale means. And that's one of the things that is clear from everything we have done over the years, and also when we have our speakers, that everyone has a slightly different view of what it actually means, right? So we talked about spatial scale, we talked about time scale. Some people really think about interdisciplinarity or, translational science, going from basic science to the clinic, see that as scaling. I originally really thought about biological organizations, or going from molecules to cells to individuals to populations to communities to the globe, but one of the things we have really found is that everyone defines it differently. So if you have anything that you think, well, this is how I think of scale, and you haven't really mentioned that, we would love to hear that.
Yeah. You can see that my own bias is on the spatial scale side, and standing 6 feet apart, was that the right distance? Is it always the right distance? Those kind of things are really interesting to me. That where something happens might tell you something about what happened. But yeah, other thoughts you might have? So in Natalie's example, there's the different types of industries that impact the landscape, and they impact in different ways. Even the roads versus a mining, logging roads versus a mining village is a different sort of impact. And it's changing the process, or changing the setting in which the process occurs. Thoughts? Or everyone's doing laundry in the background, or watching the snow out of the window? Yeah, Jon Snow. That took me a while to figure out that that's what you were talking about.
Yeah, I know. It was outside my office. I was very excited. Yeah, the other thing that's interesting, even about the spatial scale, like you just said, did the 6 feet work? Was it the right distance? And it's interesting, right? Because in the Netherlands, it was 5 feet.
Yeah.
And that was their official recommendation. And I'm wondering if that's related to the size and the scale of the country, because the Netherlands is much smaller. People are much closer to each other. So 5 feet is about all you can fit. I don't know. But it was a really interesting thing to me that everything was about that. And then here you get an extra foot.
Yeah, well, and we've all had, there's sort of cultural scales as well that can play into infectious disease. So you think about the crowding on subways in different countries. What are
Yeah.
people comfortable with, and what are
Yeah.
people uncomfortable with? One of the one of the speakers talked about Lyme disease here, and some of the vaccinated bait stations you can put in your backyard. Hopefully they'll have bait in them. It vaccinates the mice, and maybe it stops some of the cycle of transmission. But he also pointed out that the bait is in kind of a plastic yeah, oh yeah, 7 feet. Yeah, interesting. So in the bait containers, like a little house that the mice go in to feed, and they come back out. But the weasels figured out that these were like mouse dispensers to them. So they'd sit on top of them and wait for a mouse to come running out and then eat it up. So the vaccination coverage may have been high on the mouse population, but since they were eaten immediately, it was not putting out vaccinated mice into the population. It was kind of building up a pretty high concentration of the pesticide in the weasels, or the other carnivores. So ecology is fascinating because of the sort of unintended consequences, but the hubris of fixing one scale and having it go to others is like an interesting idea. Yeah, so 2 meters in Russia, maybe it's metric versus imperial distance. The pathogen is measuring differently.
But on the basis of those three countries, you could do a regression and demonstrate
yeah,
that the lower the country's density, population density, the greater the distance people had.
yeah, it does. Yeah, the Darude constant. There's some distance decay.
But the weird thing is, if you then take into consideration the skill of the people, so Dutch people are on average way taller than Americans, so they actually need more distance because they breathe at a higher level, so that they spread it over a longer distance. So that goes against any rational decision making.
Well, maybe Americans talk louder and expectorate more as they speak too. So we have
Maybe. They are loud. That's true.
they are loud, and they don't like quiet. We create conversation to fill the void.
Great.
Well, thanks everyone.
yeah.
I think I hope it gives you some things to think about. I know it's a little hand wavy in some sense, but we'll be interested to hear. Give it some thought, and next week feel free to reach out to us too if it's just something you want to chat about. Like I said, liking something about what you do and liking to talk about it is a really important part of a research career. So thanks again. Stay safe. Enjoy the snow once you're in a safe place. And we'll see you next week.
Thanks, everyone.